Hello everyone and welcome back to the BIO202 course. We're going to continue now the third and the last section of chapter 1, which talks about animal diversity. So in the previous section, we were talking about protostomes and deuterostomes and the two uh, categories of animals under each of these two kinds. Now let's talk, as I said before, that we have animals in general belong to two categories, the parazoa and the eumetozoa. So within the branch of Parazoa, there exists the phylum Porifera, as we said, which consists of sponges. And sponges are among the most abundant animals in the deep ocean, with more than 26,000 marine species and 150 freshwater species. And from their characteristics, they have many developmental forms, and most of them lack symmetry. As we said, they don't have a symmetry, they don't have tissues, they don't have organs or organ systems. The adult sponges remain sessile in the ground. That means they don't move. Okay, they always stay still, attached to the ground. And the larva, which is the newly formed sponge, if we need to say, is free swimming until, of course, it reaches a ground where it sticks to it and becomes a sessile adult and grows. Okay. Now, the functional unit in the sponge is the choanocyte cells. The choanocyte cells are the site of absorption, digestion, ingestion, or actually, it's not ingestion, filtering, if you need to say, of nutrients and gases. So what actually happens is that each choanocyte, it has this flagellum that wiggles and push water and with the nutrients and oxygen to it through the choanocyte, where absorption and digestion, or actually filtering and digestion is going to occur. Now, regarding Gymatozoa, as I said, which contains all other animals other than sponges, as we've had before, they could be either bilaterians and or nidarians. Bilaterians, which are, as I said, triploblastic. They have the three germ layers: the endoderm, the mesoderm, and the ectoderm. And then we have the other one, which are diploblasts, the nidarians and ctenophores. Okay. So let's talk first about ctenophores, the phylum Ctenophora. The phylum Ctenophora, they are also referred as comb-jellies, sea walnuts, or sea gooseberries. So they completely are found in the sea. They have a characteristic of 8 rows of comb-like plates of fused cilia that aids them in the movement. Most of them are bioluminescent. That means when they move, they produce light by a biological reaction. That's what bioluminescent mean. And they have two tentacles that are more elongated and that are covered with a coloblast. And coloblast, coloblast produce powerful adhesive chemical that allow a comb jellies to attach to the prey, okay, and thus kill it. In addition, ctenophores, they don't have nematocysts, and we're going to see what is nematocyst later, and have more complicated structure than cnidarians. Okay, so as you can see, this is a comb jelly. It has 8 rows of comb-like plates of fused cilia that helps the animal to move, and it has, as you can see, two tentacles that are covered with coloblasts that discharge a very sticky chemical, if you need to say, that allows it to attach to prey, and it is bioluminescent. So when it moves, it's going to produce a protein, if you need to say, that is going to produce light. Okay, so as we said, this is a fun idea about comb gels is that the luminescence that they show is actually it's by photoproteins, where upon the release of calcium, these photoproteins are going to get activated, and this is going to produce this magnificent light. Moving on to phylum Cnidaria, few of them live in freshwater but the majority of them are marine. Okay, they are diploplastic, as I said before, and have different tissues but lack organs. So cnidarians don't have organs. They don't have excretory, reproductive, circulatory, respiratory. They don't have any of these organs. They were the first to have muscles and nerves that resulted in their behavior, in addition to a mouth and digestive cavity. They do not have a concentrated nervous system, which implies a latticework of nerves, touch, gravity, light receptors, okay? So they only have this primitive nervous system. They are carnivores, okay, and have a unique structure called the nematocysts. Nematocysts contain specialized venom that helps them to catch prey. Okay, so the ctenophores, they have coloblasts that are sticky, that produce sticky chemicals to allow them to stick to the prey, and cnidarians had produce or had nematocysts that produce a venom that help them to catch the prey. They have two layers to the body wall, an epidermis and a gastrodermis. So an epidermis, from its name, it's the outer part, and a gastrodermis, it's a dermis, okay, that covers the gastric part, okay, the digestive tract. And we have the mesoglea that occurs between these two dermis. Cnidarians have two body forms, as you're gonna see. They can be either in the medusa form or they can be either in a polyp form. The medusa form, it has an umbrella-like shape, as you can see here, and they are free-living, they swim. Whereas the polyp form, it has this cylindrical shape, and they are sessile, okay? They do not move. Multiple cnidarians shift between these two forms as well. The body plan has a single opening leading to the gastrovascular cavity. As you can see here, this is a single opening here, and this is the single opening here. So it has this single opening. So this means that it absorbs and excretes from the same opening. So the gastrovascular cavity that is linked to this opening is used in transportation of gas. It's used for digestion, elimination of wastes, and formation of gametes in multiple species. Okay, so basically it has the most important functions. Why? Because as I said, cnidarians don't have systems. They don't have excretory system or a urinary system to excrete wastes, etc. The gastrovascular space serves also as a hydrostatic skeleton. So, as we know, for example, us humans, we have our own skeletons, our bones, in which our muscles can work on. The cnidarians don't have bones; they have a hydrostatic skeleton. It's the gastrovascular space in the water, in it, the fluid, and it acts as a if we need to say a solid state where muscles can act on. So, for example, the animal can squeeze these muscles on the hydrostatic on the gastrovascular space, just move, for example. Okay, so it has a firm structure to which muscles can operate, and also determines the shape of this animal. Okay. As I said before, it's site of extracellular digestion, which allows digestion of bigger material that sponges usually digest. Multiple polyp species build an exoskeleton of chitin or carbonate calcium around themselves. Okay, so you could build an exoskeleton. Okay, and this is mainly for protection. In multiple colonial species, a coral is a result of the exoskeleton linking members of the colony together. Some form an internal skeleton. Okay, so what is that telling us here is that the exoskeleton can sometimes some cnidarians produce this exoskeleton and form a colony. So they can deposit this exoskeleton and they form this colony to just connect all the colonies together, this exoskeleton. Okay. Also, some cnidarians forms an internal skeleton as well. Lastly, we're going to talk about acyl flatworms that belong to the phylum Acylia. They were once thought to be basal members of the phylum Platinhelminths. They have a primary nervous system. The mouth leads to a solid digestive synostium due to the absence of missing digestive cavity. So they don't have digestive cavity; they have only the mouth, and it gives the solid material to the synostom where digestion actually occur. That's it for this chapter. Thank you for coming by. Hope you benefited as much as possible. And as I said, these 3 chapters are going to be not as enjoyable as the other chapters are going to be. Thank you. Have a great day. Bye.